Hello friends, welcome to some biology class. Friends, cell death play a very important role in the development of plants and animals. It usually continues into adulthood. In a healthy adult human being, every hour, billions of cells die in the bone marrow and in the intestine. However, the tissues do not shrink because by some unknown regulatory mechanism, the rate of cell division exactly balances the rate of cell death. These normal cell deaths are suicides in which cells activate an intracellular death program, thereby killing themselves in a controlled way, a process known as programmed cell death. In animal, programmed cell death occurs usually but not exclusively by apoptosis. Apoptosis is a form of programmed cell death which is most common and best understood. Friends, today we shall discuss about programmed cell death, more particularly about apoptosis in animals. Cells undergoing apoptosis exhibit certain characteristic morphological changes which are: number 1, cells shrink and condense. In addition to this, the cytoskeleton collapses and the nuclear envelope disassembles and the nuclear chromatin condense and break up into fragments. The cell surface often bleeds, and if the cells are large, they are broken into membrane enclosed fragments called as apoptotic bodies. More importantly, the cell surface is also chemically altered so that they are easily recognized and rapidly engulfed by macrophages and other phagocytes before they can spill their content to the outside. So in this way, the cells die neatly and rapidly cleared away without causing a damaging inflammatory response. Programmed cell death is not confined to animals. It also occurs in plants and even in unicellular organisms such as yeast and bacteria. The amount of programmed cell death that occur in developing and animal tissue can be astonishing. For example, in the developing nervous system of vertebrates, more than half of many types of nerve cells die soon after they are formed. So what is the purpose of this massive cell death? Why all these cells die? Programmed cell death by apoptosis and by other processes serve several purposes. They are: in animal, programmed cell death eliminate unwanted cells by apoptosis. For example, cell death sculpt hands and feet during embryonic development. They start out as spade-like structure and the digits only separate after the cells between them die. In some other cases, cells also die when the structure that they form is no longer required. For example, when the tadpole larva metamorphoses into a frog, the tail is no longer required, so the cells in the tail die as a result. The tail disappears. In addition to this, programmed cell death also help regulate cell number. Cell death adjusts the number of nerve cells so that they can match with their target cells with whom they are connected. More importantly, programmed cell death also functions as a quality control process in development. It eliminates the cells that are abnormal, misplaced, non-functional and potentially dangerous. For example, in the vertebrate immune system, apoptosis eliminates the developing T and B lymphocytes that either fail to produce potentially useful antigen-specific receptor or produce self-reactive receptor that make the cells potentially dangerous. Apoptosis also eliminates most of the lymphocytes activated by an infection after they have helped destroy the responsible pathogen in adult tissue that are neither growing nor shrinking. Cell death and cell division are tightly regulated so that they are exactly in balance. Apoptosis occur at a greater rate in the adult human bone marrow where most of the blood cells are produced. For example, neutrophils are continuously produced in very large number, but a vast majority of them die within few days by apoptosis without ever functioning. The cycle of cell death and cell production ensure a continuous supply of short-lived neutrophils so that they can be mobilized to fight an infection. In animals, cells can recognize if there is any damage in their organelle, and if the damage is great enough that they can't repair, they kill themselves by undergoing apoptosis. For example, DNA damage causes mutation, and you know that some of the mutations are cancer promoting. Cells have various ways of recognizing DNA damage. They also have specific mechanism to repair them. But if the cells fail to repair this DNA damage, they often kill themselves by undergoing apoptosis. In addition to morphological changes, cells undergoing apoptosis also exhibit certain characteristic biochemical changes which are: number one, in apoptotic cells there is cleavage of chromosomal DNA. The chromosomal DNA is cleaved by an endonuclease into fragments of distinctive size. In cells undergoing apoptosis, an especially important biochemical change also occurs in the plasma membrane. The negatively charged phosphatidylserine, which is normally exclusively located in the inner leaflet, flip to the outer leaflet where it serves as a marker of apoptosis. In addition, the electrical potential which normally exists across the inner mitochondrial membrane is often lost in apoptotic cells. Finally, in cells undergoing apoptosis, there is relocation of cytochrome C. Cytochrome C is a member of electron transport system. This protein is normally located in the intermembrane space of mitochondria. In cells undergoing apoptosis, cytochrome C is released from intermembrane space of mitochondria to the cytosol where it serves as another marker of apoptosis. Now we shall discuss about the mechanism of apoptosis. Apoptosis depends on an intracellular proteolytic cascade mediated by caspases. Caspases are a family of proteases that have a cysteine residue at their active site. They cleave the target protein at specific aspartic acid. Because of this, they have been named as caspases. C for cysteine and ASP for aspartic acid. Caspases are synthesized in the cell as inactive precursor known as procaspase. These procaspase molecules are the proenzymes which are activated by proteolytic cleavage. Procaspase cleavage occur at one or two specific aspartic acid residue mediated by caspases which are already active. So friends, this figure explains the activation of procaspase. These are the two molecules of procaspase. These are the two prodomains. This is the amino terminus and these are the carboxyl terminus. So these procaspase molecules are cleaved by the caspases which are already active. In this process, the procaspase molecules are cleaved into a large subunit and a small subunit. In this process, the prodomains are usually removed, and the large subunit and the small subunit together form a heterodimer. Two such heterodimers assemble to form an active tetramer. Once activated, the caspase molecules cleave and activate other procaspase molecules. From the earliest stages of animals' development, healthy cells continuously make procaspases and other proteins concerned with apoptosis. So the apoptotic machinery is always in place. All it requires is a trigger to get activated. So how the caspase cascade is initiated and more particularly how the first initiator procaspase is activated. Initiator procaspases have a long prodomain that contains a caspase recruitment domain that enables them to assemble with the adapter protein to form an activation complex. After incorporated into such an complex, the initiator procaspases are brought into close proximity that is sufficient to activate them. So they cleave each other, thereby making the process irreversible. The initiator caspases then cleave and activate the downstream executional procaspases that results in initiation of the caspase cascade and amplification of the death signal throughout the cell. Procaspases operating in the process of apoptosis are divided into two different groups. They are initiator procaspase and executioner procaspase. Initiator procaspases operate at the start of apoptosis. These are the two molecules of initiator procaspase. These are the prodomains. Each molecule of initiator procaspase is cleaved by an active caspase into a large subunit and a small subunit which together form a heterodimer. Two such heterodimers assemble together to form one molecule of active initiator caspase which then cleave and activate the downstream executional procaspase which then activate other executional procaspase. So the executional procaspases are activated into executional caspase which then cleave specific target proteins in the cell that include nuclear lamins, components of the cytoskeleton and cell-cell adhesion proteins. Cleavage of nuclear lamins by executional caspase leads to an irreversible breakdown of nuclear lamina. Another target of executioner caspase is a protein that normally holds the DNA degrading enzyme in inactivated form. Other targets are components of the cytoskeleton and cell-cell adhesion protein. Cleavage of these proteins help the apoptotic cells to round up and to detach from neighboring cells, which make it easier for the healthy neighboring cell to engulf it. From the earliest stages of animals' development, healthy cells continuously make procaspases and other proteins concerned with apoptosis. So the apoptotic machinery is always in place. All it requires is a trigger to get activated. So how the caspase cascade is initiated, and more particularly how the first initiator procaspase is activated. Initiator procaspases have a long prodomain that contain a caspase recruitment domain that enable them to assemble with the adapter protein to form an activation complex. After incorporated into such a complex, the initiator procaspases are brought into close proximity that is sufficient to activate them. So they cleave each other, thereby making the process irreversible. The initiator caspases then cleave and activate the downstream executioner procaspases that results in initiation of the caspase cascade and amplification of the death signal throughout the cell. Friends, there are two different pathways which can activate the proteolytic cascade leading to apoptosis in mammalian cells. These pathways are number 1, the extrinsic pathway, and number 2, the intrinsic pathway. Both of them are best understood. First of all, we shall discuss about the extrinsic pathway of apoptosis. The extrinsic pathway is activated by the cell surface death receptor. The binding of extracellular signal protein with the death receptor trigger the extrinsic pathway. The cell surface death receptors are transmembrane proteins. They have an extracellular ligand binding domain, a single transmembrane domain, and an intracellular death domain which is required to activate the apoptotic program. The cell surface death receptors are homotrimers. They belong to the tumor necrosis factor receptor family that include a receptor for tumor necrosis factor itself and the fast death receptors. The ligands that binds with the cell surface death receptors are also homotrimers. This figure shows the activation of extrinsic pathway of apoptosis. A well understood example of activation of cell surface death receptor is the activation of fast death receptor present on the surface of a target cell by the fast ligand present on the surface of a cytotoxic T lymphocyte. This is a cytotoxic T lymphocyte which has fast ligand on its surface, and this is a target cell which has fast death receptor on its surface. As already mentioned, the cell surface death receptors are homotrimers. They have three different domains. These are the extracellular ligand binding domain that binds with ligands, and the transmembrane domains are embedded in the plasma membrane, and these are the intracellular death domain which is required to activate the apoptotic program. This pathway begins with the activation of fast death receptors by the fast ligand. When activated, the fast death receptor recruit the intracellular adapter proteins which in turn recruit the initiator procaspases 8 or 10 or both and forming a death-induced signaling complex known as DISC. When activated in the death-induced signaling complex, the initiator caspases cleave and activate the downstream executioner procaspases, leading to the apoptosis of the target cell. There are many cells which produce inhibitory proteins that act directly or indirectly to restrain the extrinsic pathway of apoptosis. For example, some cells produce decoy receptors that only have the extracellular ligand binding domain but not the intracellular death domain. Such decoy receptors can bind with the ligand but cannot activate the apoptotic program because they do not have intracellular death domain. So, with the help of decoy receptors, the cell surface death receptors are competitively inhibited. So, friends, this is all about the extrinsic pathway of apoptosis. In addition to the extrinsic pathway, the apoptotic program can also be activated by the intrinsic pathway. The apoptotic pathway, which is activated from inside the cell without the involvement of cell surface death receptor, is called as the intrinsic pathway. It usually occurs in response to cell injury or other stress, such as DNA damage or lack of oxygen or nutrients. In vertebrates, such intracellular activation of the apoptotic program occur by the intrinsic pathway, which depends on the release of mitochondrial protein from the intermembrane space into the cytosol. When released into the cytosol, some of these mitochondrial proteins activate the proteolytic cascade that leads to the apoptosis of the target cell. One of the key mitochondrial protein that activate the intrinsic pathway of apoptosis is cytochrome C. Cytochrome C is a water-soluble component of the mitochondrial electron transport system. This protein normally resides in the intermembrane space. When released into the cytosol, cytochrome C activates the intrinsic pathway. This figure explains the activation of the intrinsic pathway of apoptosis by cytochrome C. This is a mitochondrion. This space is the intermembrane space which contains cytochrome C. When released into the cytosol, the cytochrome C molecules binds with the initiator procaspase activating adapter protein known as APAF1. This is the initiator procaspase activating adapter protein known as APAF1. The binding of cytochrome C with the APAF1 leads to the oligomerization of APAF1 into a wheel-like octamer known as apoptosome. This is a complex known as apoptosome which has been formed due to the oligomerization of APA-F1 molecules. In the apoptosome, the APA-F1 molecules recruit the initiator procaspase 9. The initiator procaspase 9 are activated by their close proximity. After activation, the initiator caspase 9 cleave and activate the downstream executional procaspases that leads to the apoptosis of the target cell. So friends, this is all about the intrinsic pathway of apoptosis.